Hi, my name is Jocelyn Rigo, and I'll be presenting my work done alongside John Carter, Daniel Schwartz, Vegas Bandawat, and Edward Kim, focused around learning spiking neural network models of Drosophila olfaction. Here, we investigate the Drosophila olfactory system using biologically inspired computational modeling methods, like spiking neural networks and leaky integrate and fire neurons. In an effort to learn more about the mechanisms of low-level perception, we observed the neural architecture of these circuits and aim to replicate an in silico architecture that resembles them. Specifically, here we investigate the mechanisms of olfaction at the level of first to second order neurons. We chose to model Drosophila olfaction as opposed to a system in another model organism because the central nervous system of Drosophila is relatively small, with roughly 100,000 total neurons in the brain. The Drosophila system is thus at a scale that can be modeled with computational methods. The figure here shows a sketch of the Drosophila olfactory system, which contains about 1,300 olfactory receptor neurons. Odors are detected by these receptor neurons, which project to second-order projection neurons. Projection neurons further project to the mushroom body and lateral horn neurons. The connection between olfactory receptor and projection neurons is the particular focus of our study. Our lab built a novel apparatus in which flies explore a half-meter wide circular arena. Odors were delivered with an olfactometer producing a continuous stream of air directed over the fly. Tenfold diluted odors were presented to flies at about 8 seconds. Whole cell spike recordings were obtained from both olfactory receptor and projection neurons over 10 20-second trials, resulting in timestamps of spikes. We transformed this data into spike counts over 50 millisecond bins with 25 milliseconds of overlap in order to process it with rate-coded models. Here shows the spike timestamp data for 10 trials of 20 seconds. As you can see, at around 8 seconds, there's a large increase in spikes at the odor onset, followed by a decrease in spikes. The first rate-coded model employed was a Gaussian generalized linear model, which has been shown to be particularly successful in modeling neuron activity in response to sensory stimuli. Using our rate-coded data, we modeled connections mapping 10 ORNs to each of our 10 PNs separately. The resulting coefficients from these 10 models were then averaged together, resulting in a final 10 coefficients representing the average impact of each ORN on the activity of a PN. Next, we employed a Poisson generalized linear model, which has been shown to resolve some flaws of the general linear models. We used a Poisson GLM with a log length function to similarly model the effect of the activity of 10 ORNs on each PN. The coefficients of the resulting models were then averaged together, producing 10 coefficients like our Gaussian GLM model. As shown on the right, our Poisson GLM was fairly successful at predicting the spiking activity of projection neurons, especially in predicting the increase in activity shortly after odor onset. Next, we moved away from rate-coded models towards neuromorphic implementations that use local learning rules to inform synaptic connections in an attempt to learn directly based on spike input. In neuroscience, the neural engineering framework is a set of principles for constructing networks based on biological constraints. In order to construct our models based on this framework, we use the Nango software module. We built our model in Nango using the biologically plausible supervised learning rule called prescribed error sensitivity. This PES rule adjusts connection weights between neuron populations to minimize some error over time. The equation here shows how the change in sum weight, W sub ij, is calculated, where K is a scalar learning rate, N is the number of neurons in the presynaptic population, A sub j is the gain of the postsynaptic population, E sub j are the encoders of the postsynaptic population, and E is the error to minimize. We initialized 10 Nango nodes with our recorded ORN spike trains as input. These 10 nodes were connected to a single leaky integrate and fire neuron, representative of a projection neuron. This model attempted to learn the connection weights of the 10 ORNs to the observed PN with the described PES learning rule. Next, we used Intel's Loihi neuromorphic chip to model the ORN to PN connection. Our network was built using 3 primary objects: compartments, spike generators, and learning rules. Compartments in Loihi allow for voltage to integrate bias and spikes, and spike when this exceeds a specified membrane potential. Following a spike, this voltage resets. Spike generators generate spikes from our ORN spike trains. We chose to use a spike timing dependent plasticity learning rule to update our model's weights. The equation here shows the form of STDP learning rule used, which updates some weight by pre-X and post-Y spikes. This model has been shown to reproduce key features of long-term potentiation and long-term depression. For our experiments, we modeled the connection of 10 ORNs to a single PN. Given the weights learned by each of our different models, we simulated spike traces for a projection neuron. We then compared these predictions to actual collected PN traces. Here, we show an example spike train generated by each of our different methods, as well as 2 trials of actual PN data in black. Noticeably, these 2 actual spike trains show the variability that exists in real-world recordings of responses to the same stimulus. In order to quantify our results, we used a measure of spike synchrony called the interspike interval metric, which evaluates the ratio of instantaneous firing rates. Our results are shown in the table on the bottom right of this slide. Our data shows that most methods model spike trains well during pre-stimulus, 0 to 8 seconds, and presentation of stimulus, 8 to 9 seconds. Our adaptive Loihi implementation outperforms other methods during adaptation, 9 to 11 seconds, due to activity range homeostasis. Our work effectively investigates several learning implementations of increasing biological plausibility for modeling Drosophila olfaction at the level of olfactory receptor neurons to projection neurons. Our results show that most methods model spike trains well during pre-stimulus and during stimulus timeframes, while the Loihi implementation surpasses other methods post-stimulus during adaptation. Thank you.